Hello, my name is Eva Faddoul. I am a rising senior at Regina High School, and I was able to do my research in Dr. Zong's lab. And this is the examination of the promoter activity of the mouse S-SOX5 gene in IMCD3 cells. So what is the mouse SOX5 gene? This gene is involved with the regulation of embryonic development and in the determination of cell fate. Mouse SOX5 gene encodes two major isoforms, L-SOX5 and S-SOX5. These two SOX5 proteins have different tissue distribution. And these are the different tissue distribution of two SOX5 isoforms. So these two variants are the L-SOX5, which is the longer version, and this is the S-SOX5. What's interesting about this figure is in the S-SOX5, you see that the first exon is not transcribed. And then figure B and C are what the L-SOX5 and S-SOX5 and what tissues you can find them in. So what is the big picture? One of the many projects in Dr. Zong's lab is to study the transcriptional regulation of this S-SOX5 gene. In my project, I was able to amplify the SOX5 gene promoter region and subclone it in the PGL3 vector and transfect them into IMCD3 cells. This is the strategy to study the regulation of the S-SOX5. So number one is the longer version of the S-SOX5, L-S-SOX5. And number two is the shorter version, S-SOX5. As you can see, there's less base pairs that we studied. So the PGL3 vector, why did we subclone this promoter region in the PGL3 vector? The PGL3 vector has no promoter region. This allows us to add our own S-SOX5 promoter region to the vector. The PGL3 vector is used in our case because the modified firefly luciferase in the cDNA encoding. If the luciferase is transcribed, this means that the regulatory element is active. And this is where the luciferase is. So it will light up in a Western blot or in a luciferase assay system. This is my flow chart. Oh, missing one. I drew this flow chart. We started with the PCR, we did TA cloning, then we did plasmid isolation and restriction digestion, and then we subcloned it into the PGL3 vector, and then I don't know why it didn't show up, but restriction digestion did it one more time, and then we transfected it into cells, and this is what it would look like. It's a little diagram what it would look like if the luciferase is active. Oh, there it is. Okay, we started with the polymerase chain reaction. The PCR method is used to make billions of copies of a specific DNA sample. This allows the S-SOX5 DNA to be amplified. And we used the S-SOX5 DNA to do a PCR, and it made many copies as shown. Next is TA cloning. This method is used for the cloning of PCR products. The TA cloning will clone the promoter region of the S-SOX5 gene to a vector. The vector that was used is a topo 2.1 vector, which is used for high amounts of DNA. And as you can see, these were the results. You grow them on a bacteria dish because bacteria, they grow faster. Next is plasmid isolation and restriction digestion. The bacteria from the TA cloning were picked up and the plasmid DNA was isolated from the bacteria. The method of restriction digestion was used to cut specific sites that surround the DNA sequence. The enzyme EcoR1 is used to cut the DNA at specific locations. One can gain indirect sequence information from the presence and location of the insert. So as you can see, we digested just the PGL3 vector and then SOX5 PGL3 1 and 2, which was just two different copies of the same primers. Sequencing. The samples of the plasmid DNA were sent out to a company to read the base pairs. There had to be no mutations made in the DNA. And this is what it would look like when they send it back to us. My mentor, Candace, had to read each of these letters and make sure nothing was mutated, which is really hard. I tried it with her. Subcloning. Subcloning is used to move a DNA sequence from one vector to another. the transfer of the SOX5 DNA plasmid to the PGL3 vector. And again, we did it in bacteria plates because it grows faster. But this is where we put the SOX5 DNA into the PGL3 vector. Next, we had to do plasmid isolation and restriction digestion again. Once again, the plasmid DNA was isolated from bacteria. When restriction digestion was performed the second time, the enzymes NHE1 and XHO1 were used to cut the DNA. So obviously the SPINK2, SNAP-in, 790, and SNAP-in 807 were used for different projects, but these two were the what we digested and isolated. Next is cell transfection. Cell transfection is the introduction of foreign DNA into a cell. IMC3 cells are commonly used for studying cilia. These cells are kidney cells. The cells grow on a plate, and after 1 to 2 days, one would lysate these cells. Cell lysis allows the cell membrane to open and the luciferase can be released. I drew this diagram just to see what the plates looked like. So obviously one was non-transfected for the control, and the one was PGL3 vector, just the PGL3 vector. And then next is SOX5 PGL3-1 and SOX5 PGL3-2. And these were sequenced perfectly. There was no mutations. Luciferase activity. The luciferase assay allows one to study the regulatory control of interest. If the luciferase was transcribed, then the regulatory element of the gene is active. This can be done in a luciferase assay. And this is our results. So we did it correct. There was no mutations, and the SOX5 gene was able to be transfected into the cells with the PGL3 vector. And you can tell because the higher the number, the more active the PGL3, the SOX5 DNA was. So SOX5-1 worked and SOX5-2 worked. This is great. In conclusion, luciferase activity can be shown when the promoter region of SOX5 gene is amplified and transfected into IMCD3 cells using the PGL3 vector. Acknowledgement, a big thank you to Dr. Zong and Dr. Lee, and a major thank you, a ginormous thank you to Candace for teaching me everything. Thank you to Dr. Moore, Dr. Alvaro, thank you, Alyssa, and thank you to all the other members of our team, including Eva. Thank you. Thank you.